Good morning, everybody. I'm just a little behind the ball here. That's where we ended up. Okay, just making sure I know where we were at. What am I saying? Don't know why that defaulted to my stamps.com page. Okay, so today we're going to get into the weeds of bacterial gene regulation. Where were we? Okay, so we ended up finished about here. Sorry, kind of a little awkward because I've got a dog at the end of my foot. And basically, I think what we covered at the end of the last class is this idea that essentially there are two parts to each operon, right? We have the operon itself, which contains the structural genes. There, I need some water. And that operon consists of the structural genes and the promoter operator, right? The promoter is where the RNA polymerase binds. The operator is where the regulator protein may or may not bind, depending on the context. Context is everything when it comes to controlling gene expression. There's also a gene encoding the regulator protein, and that might be right next to the operon. It may be somewhere completely else. And that's expressed all the time at a low level. The operon may or may not be, right? Hence the control of it. And this protein may or may not be able to bind that operator, again, depending on whether it's an inducible or repressible system. Here's a quick question. So in the absence of anything else, if this was an inducible system, would this protein bind? If it's an inducible operon and nothing else bar the regulator protein is present, would that regulator protein bind? A little bit of revision and recap for you. Think about what the outcome of an inducible system is. What is it that you're doing to turn it on?
Can you repeat the question?
So I'll type it up too, that will help. So if this is an inducible operon and only the regulator protein is present, will it bind the operator? So this is another example of asking you the same question, but in a different way, kind of thing that I'll try and do in the exam. So it's kind of like a 50-50 answer, right? You've got 1 in 2 chance getting it right. Well, how about the other answer?
Yes.
Okay. Yes, because in an inducible system, you have to induce the repressor to leave, right? So you got to think about what's the outcome for an inducible system, right? So if the regulator protein on its own is present, it will bind to the operator. So the answer is yes. In the absence of the inducer. Now, when the inducer is present, that then results in that regulator protein leaving the operator, and so the operon is expressed. So the operon is induced to be expressed, right? It's inducted. Not sure that's the right word. Always think of the Hall of Fame for various things when I hear the word inducted. Now, if this is a repressible operon and only the can't see what I'm typing here. Only the regulator protein is present, will it bind the operator? Same question, now different context. So this is a repressible system. So the outcome is that the operon is turned off. It is repressed. Now, is this a yes or a no?
No.
Yay, good job, everybody. It's a no, because in a repressible system, that's actually called an caught myself there. What's the repressor called? What's the repressor alone called in a repressible system? Yeah, good job, Jonathan. Awesome. Exactly. That's exactly what it's called. So what do you need to be present for the aporepressor to be able to bind the operator? Exactly. You have to have the co-repressor. So now you can see the context and kind of how this system works is super important in terms of the outcome, right? And really, at the end of the day, it's the outcome that we care about, right?
So the co-repressor or the aporepressor needs the co-repressor?
Correct. So
Okay.
those two together are now able to bind the operator. And it's all about protein shape. So we talked about a little, oh, hello there, Jesus. A little while ago, we talked about how proteins can recognize DNA sequences, right? Specific sequences of A, C's, G's, and T's. They have to have the right shape to do so. And so the deal here is an aporepressor does not have the right shape to recognize the operator. Only when the co-repressor is bound does it adopt the correct conformation. That's why it needs the co-repressor, because in the absence of it, it won't recognize the operator. And in repressors which are inducible, so they can be induced to leave, it's the opposite. So the outcome is opposite, the mechanism is opposite. So in the absence of an inducer, the repressor will bind and have the correct shape to bind the operator. When the inducer binds to it, now the shape changes and now it doesn't recognize the operator. So it's essentially, you know, the two sides of the same coin. They're opposites, really. And you always have to kind of keep in mind the context of what's going on. Like, what is the need for the output of this particular operon? Okay, anybody got any questions? Give me a little puppy shot. Oh, she's not a puppy anymore. Where is she? There you get to see my foot. Now I can stroke my dog.
So the aporepressor is always there and the co-repressor comes to find it, basically.
Yes, that's exactly
Okay.
right.
Thanks.
Yeah, so both the aporepressor in a repressible system and the repressor in an inducible system are always present. Yeah. it's either the inducer or the co-repressor, which may or may not be present, and that's what determines whether or not they bind to the operator. Good question. Okay, so we're going to move on to the lac operon. And before we do that, I want to really talk about like a bigger picture concept, right? So we've already talked about this general structure, right, that you see in front of you. The operon with its regulatory region and its structural genes, and then the regulator gene, which is kind of always expressed at a low level, right? It's not really controlled. In fact, you can even tell that because there's no operator there, right? So this regulated protein will always be produced, right? Always present. Now, bacteria have, I was trying to think of a good analogy yesterday and I kind of didn't. My best analogy, I haven't come up with a better one, is it's like when you're really, really, really hungry and you sit down for dinner, right? I haven't eaten anything all day. Sit down for dinner and I don't know about you, but I'm like a real foodie. I'll just scarf it. Like eat as quickly as I can because I'm starving and I don't like being starving. It's not good. But then once I start getting full, I'll be like, oh, whew, feel better now. Less grumpy, not shouting at the kids. Oh, being grumpy at the kids. There you go. That's a better thing to say. And I'll slow down and I'll just kind of enjoy my food a little bit more, right? So it's essentially a similar kind of deal with bacteria, right? So they have a need, if they need something and they don't have it, they want to produce a lot of it really quickly, right? And to do so, if that something is produced by the proteins in an operon, they'll turn up expression really high, right? So you'll get lots of expression, you'll get lots of proteins produced, you'll get lots of whatever product is that the bacterium needs, right? But this is a really expensive deal, right? Transcription, and especially translation, costs a lot of money in terms of energy. So you need energy, but you don't want to spend more of it than you have to. It's kind of like money in a sense, right? And so once the bacterium kind of satisfies its craving, essentially, then it goes into a kind of a more measured like maintenance phase, right? I've got enough of whatever product I need. Now I just need to make sure that I keep making enough of it to cope with whatever my needs are, right? And so there are two kind of complementary but different mechanisms by which bacteria control gene expression. The first one I often kind of refer to as like Frankenstein's monster switch, right? You know those big kind of blade switches. It's like lightning, brrr, Frankenstein's monster, that kind of thing, right? It's a big on-off switch, right? Very little expression. I mean, there's always a little bit, but it's really, you know, barely any, to gonzo balls amounts of expression, right? And so it's a kind of a very binary deal, right? It's either on or it's off, right? Now that works when you need a lot of something very quickly, but it doesn't work very well when you need to kind of, you know, nuance the deal, right? And so bacteria also have kind of more of a dial, which I always remember TV ad by the Wallace and Gromit creator, Nick Parks, called about a couple of turtles with natural gas heating. And they go, ooh, it's lovely. Turn it off and on again, like that, in my best Yorkshire accent. And so it's kind of like more of a dial, right? So you can tweak the level of expression to just maintain what you need, but not either spend too much in terms of gene expression or run the risk of running low, right? It's kind of like homeostasis, like how we modulate our body temperature or blood glucose levels, stuff like that. They have the same kind of idea essentially. Right? And so keep that in mind, because for both of the examples that we're going to talk about, the lac-operon and the trip-operon both have those two mechanisms, a big on-off deal and a kind of intermediate sort of deal. Okay. And again, there's a lot of details and mechanisms in here. So really try and keep in mind, at least this is what works for me, the bigger picture as we go through this, and try and slot in these different things into that bigger picture. It'll make it a lot easier to cope with, right? There's a lot of stuff to absorb. Okay, so the lac-operon, anybody guess what that's involved in metabolizing? What begins with lac that you might find in the environment? Actually might go, yeah, lactose. It's like what my daughter doesn't like, actually. So she'd not be a very good bacterium. Right, so it's all about metabolizing lactose. Now lactose, bacteria, if they had the choice between glucose and lactose, they'd be like, oh, don't like that, I want that, right? And so the preferred energy source of bacteria is glucose. I'll just tap that down. And largely because it's easier to metabolize or easier to turn into ATP. And just like us, ATP is the energy currency of bacteria, right? They need it to do all kinds of stuff. Well, pretty much everything really, apart from, you know, GTP, but GTP is made using ATP in many cases, right? So if they have glucose present, they will, I don't know, keep switched off. That's clunky language, unfortunately. Any operon that is involved in metabolizing different energy sources. So, you know, again, I'm not sure the king of terrible analogies, but certainly a good practitioner. My analogy always about food, you know, because I'm, you know, love eating. And it'd be like, you know, I love curry, right? Grew up in the UK, curry is a big thing, went to university in Birmingham, where curry is even bigger thing. And very big Indian population, or yeah, Indian population in Birmingham. Anyway, it's like, you know, if I had a choice between curry and fish and chips, I'd choose curry every time, right? Or maybe like most of the times. So essentially there are all these other kind of cookbooks, right? You know, there's like, you know, fish and chips, there's Italian food, pasta, I don't know, Asian, whatever, American, how to grill a burger, that kind of stuff, right? So all of those will be off if my preferred food source is present. However, if that food source is not present, I still need to eat, right? And then I go scrounging around through the fridge, seeing what else I've got around to make dinner with, right? And if I say I have some tomatoes and some onions and, you know, garlic and maybe a bit of beef, I'd probably pull out the pasta cookbook, right? And so it's a conditional deal. If the preferred food source is not available, the bacteria has a whole range of cookbooks for turning other food sources into glucose. And the lac-operon is one of those. So you only need it when you have no glucose and you have lactose. If you have no glucose and no lactose, the lac operon is still no use, right? Maybe there's fructose present, right? You'd use the frucoperon, maybe. I'm not even sure what that's called. I'm sure there is one. Or whatever. Like if there's trehalose present, you'd use the treoperon, and so on and so forth, right? So keep in mind that context, right? Lac operon is an inducible system because most of the time the bacteria does not need it.
This slide that we're looking at is the overall picture of the lac operon.
No, this is the overall picture of a generic kind of, I haven't gone on to the lac operon, sorry. Got a little bit sidetracked with my analogies. No, this is just a generic picture of an operon. The lac operon, yeah, looks like so. Same kind of deal, right? So you can see the same parts there. Maricela, I'll get to your question in a moment. Right, we have the repressor gene, right, which is what we're talking about here, which is the regulator. In this case, it's actually right next to the lac operon. We have the promoter, where RNA polymerase binds. We have the operator, where the repressor binds, and then we have our structural genes. Quick answer to Maricela. Pozole, I'm actually not sure I know what that is, to be honest. I'm not like 100% on Latin American foods in terms of knowing what they are. Menudo, actually, I really like. That's one of my wife's favorite dishes. She's from Ecuador. So, you know, some nice rice and lentils with a fried egg on top. Super yummy. And caldo, that's like kind of beef, right, of some sort. Anyway, that sounds good to me. To be honest, I'll eat pretty much anything that doesn't have peanuts in it. Really, anything, actually. I once ate termites when I was in Venezuela. There was like some termites. They tasted like lemon. Not sure I'd want to have to live off them, but, you know, it was interesting to try. So anyway, got to get my focus back. Oh, Kiana, that's a long, long question. I'll have to tell you that another time, maybe in lab. That's why the, where'd my, God dang it. Oh, I can't see. But anyway, if you look at my YouTube channel where I post all these, my username is The Wandering Professor. So it's kind of, I've been all over the place. What is it? I've lived in 5 countries on 4 continents, I think is the current tally. Most likely not going to increase from there. So these structural genes, these are all involved in the metabolism of lactose, right? And so to do so, we need to do a couple of different things. One, we have to get lactose into the cell, right? So that is encoded by LAC-Y, right? So this encodes what's called a permease, because lactose is pretty bloody large, you know, in the grand scheme of things. I mean, it's not as big as a protein, but a disaccharide is a fairly chunky molecule. And so we need a specific transporter to get that lactose into the cell. There's no point, if you're on lactose and you can't get it in, you still can't metabolize it, right? And then once it's in the cell, we've got a couple of other proteins, one of which is called beta-galactosidase, which is a bit of a mouthful. And essentially that turns lactose into galactose. And I have no idea what happens to that. And glucose. Actually, I should look that up because I can't keep telling my students I don't know what it ends up as. But anyway, the thing that we care about is glucose, right? And so there are 2 kind of signals here. Is lactose present or not? And how much glucose is around, right? So kind of keep that in mind. We've got the beginning of the process or the substrate, and we've got the product. Both of those are going to be important in the control of the lac operon. Yep. So in addition to that, we have some regulatory elements. So here's just a slide essentially describing what I just said. We've got our repressor gene, the LAC-I gene, and we have the promoter and the operator, LAC-P and O. Okay, so again, kind of fitting with the context, if there's no lactose present, there's no point expressing the lac operon, right? It'd be a complete waste of energy and time. So in the absence of lactose, LAC-I, this repressor protein, will bind to the operator, right? So the default state of the lac operon is off. And I'll type that out because that's important. Due to the presence of the LAC-I repressor on the LAC operator. And so this state will pretty much continue until lactose is present, right? And so you're probably wondering, well, you know, if we need LAC-Y produced, how could we have lactose getting in the cell to be sensed, right? Well, essentially, the kind of like slightly dirty secret is that this LAC-I protein doesn't always stop RNA polymerase from transcribing the gene. So there's a very, very, very low level of transcription from the LAC operon. Very, very low. So you have just enough of this LAC-Y produced to be able to detect lactose being present in the environment. But in the terms of what context of what we're talking about, the LAC operon is off. Now, if you are on lactose as a bacterium, then you want the option to be able to express this LAC operon, right? So no, we haven't talked about glucose yet. We're going to talk about that in a bit. And so when lactose is present, now the repressor LAC-I can be induced to leave. And so when LAC-I isn't bound to the operator, now we get transcription and we get the production of these three proteins. So this is our big, you know, on-off switch, right? No lactose present, off. Lactose present, on, right? Very binary kind of deal. Now, what about if glucose is present as well? Now we've got our like slightly more complex kind of question, right? So bacteria have the preference for glucose because, as you can see, it takes a lot less work to get glucose than it does to get from glucose than it does from lactose, right? So now if you have glucose and lactose present, the bacteria, so lactose is present, right? So the LAC operon can be on. But now, well, it can be on, but we don't really want it to be on, right? Because we have glucose present. So now we have to find some way of dialing back the LAC operon when there's glucose present, right? So essentially there's kind of two extremes that we've got here in terms of output. So the first extreme is glucose present, lactose absent. And that equals next to no expression, essentially off. Right, so the bacteria have what they want and there's no alternative food source. So it's fine, right? You know, bacteria's like, yeah, like I'd care. And it's happily munching away on the glucose. So that's one extreme. See, talking about extremes, there has to be the other extreme as well. So the other extreme is glucose absent, lactose present equals massive amount of lac operon expression. I didn't put that in number one, that it's lac operon we're talking about, but I figured you get that. So those are two extremes. Preferred food source, no alternative, no lac operon expression. No preferred food source, lactose present, lots of gene expression from the lac operon. Because we need to make that glucose because the bacteria needs it to be able to do whatever it needs to do as a bacterium. Okay, so that will make sense. Okay. So you're probably thinking about the whole, well, what lies in between, and also where does glucose come in? Right? Because all we've really talked about so far is, in terms of the lac operon expression, is lactose present or not, right? So where does glucose fit in? And so glucose is part of that fine tuning mechanism. Either turn it up or turn it down. And so you also have, pardon me, an intermediate. And this is anywhere between those two extremes, right? We're not talking about it's like halfway in between. It just depends on glucose concentration. And that's when lactose is present, but there's also some, not a lot, of glucose present. Well, I mean, not a lot, but a variable amount. And so the bacterium needs some way of detecting glucose levels. And this is done via an intermediate, well, not even, no, intermediate's not quite the right word. A secondary metabolite called cyclic AMP. And so if you think about this too, right, as, let's go back to this picture here. As lactose is brought into the cell and it's converted by the products of the lac operon into galactose and glucose, glucose levels are going to increase. Right? If you think back to the whole terrible analogy of eating your dinner, right? Now your level of satiety, whatever the word is, starts going up, right? So glucose starts increasing. It's still maybe not enough, but it's enough so that you kind of got over that initial, I'm starving. And now you need to just start dialing back the expression from the lac operon. Because you still need lactose being turned into glucose, but you don't need quite as much, right? Also consider that not only do we have glucose being made by the products of the lac operon, but also we have glucose being consumed by the bacterium, right? So now we start getting into that kind of homeostasis deal, right? So maybe a bacterium has a kind of a flagellum, right, which takes an enormous amount of energy to work. They're really cool though, right? And so say the bacterium wants to hoof it some other part of, I don't know, the cat's milk dish, whatever it might be at. And it's going to burn some AT to kind of spin its way over there. So glucose needs to be turned into ATP, glucose levels drop. Now we need to dial up the lac operon a little bit, right? And so it's really about kind of maintaining the level of glucose that we want. And it's going to swing, right, from one extreme to the other, potentially, depending on how much glucose is being made and how much is being used, right? And so that's done by sensing cyclic AMP levels. So glucose can't be detected directly. You can't kind of just go, oh yeah, got enough. You have to detect another metabolite instead. And this is what we do too. So not necessarily when you as an organism is starving, right? Because that's a different set of pathways and sensory mechanisms. But in individual cells, when their glucose starts being depleted, we use exactly the same metabolite. So glucose levels are detected indirectly by levels of cyclic AMP. And essentially, it's an inverse relationship. As glucose, gotta be able to spell that right, it's not that hard of a word, levels increase, CMP levels decrease. As glucose levels decrease, you guessed it, cyclic AMP levels increase. So it's like a teeter-totter, essentially, like a playground swing, not a swing, seesaw. There you go, that's what we call them in the UK. Call them teeter-totters in the US for some reason. So as one goes down, the other one comes up, and it's essentially this kind of balancing act between the two, right? And so really to detect the levels of glucose, what you're doing is you're detecting cyclic AMP levels instead. And so there has to be a mechanism to detect cyclic AMP, right? That's where things start getting complicated. And so let's use a picture, it's a lot better. So this is a little bit, holy crap. So what I'm going to do is just block that off. So I don't terrify you too much, right? And so essentially, cyclic AMP is detected by another protein called the catabolite activator protein. That's what this thing in blue is. Catabolite activator. I need to touch up on my touch typing. And the way that glucose levels or lack of them feeds into lac operon expression is that when there's a lot of cyclic AMP, it binds to CAP. And that complex helps RNA pol bind to the LAC-P promoter. So LAC-operon expression can occur because there's no LAC-I bound to the operator. Right, so we've switched the operon on. But now we need more help to get the RNA polymerase binding to the LAC-P promoter efficiently. I remember we're talking about different efficiencies of promoters depending on the sequence of like the -10 and -35 boxes. We're talking about gene structure, the structure of promoters. Well, the LAC-P promoter is not a very good one. And so the RNA polymerase needs a bit more help to bind to that promoter so it can start transcription, right? And that help is provided by this CAP-CAMP complex. All right, so now we have lactose present equals no LAC-I bound to LAC-O operator, right? Glucose low or absent equals high cyclic AMP. So this is like a logic process, right? Where you work through the different steps essentially to get the output. High cyclic AMP equals lots of CAP-CAMP or CAMP-CAP, whatever you call it. Equals very efficient RNA pol binding equals lots of LAC-operon expression. Okay, questions? There's a lot to take in, I know that. Appreciate it.
So is it good to have high CAMP?
If you don't have lots of glucose, then you will have high cyclic AMP because they just work in an inverse relationship. So it's not really about what's good or bad, it's about optimizing the LAC-operon expression depending on glucose concentrations. And so if glucose is low, essentially we're get you back up there. Right, if you don't have that, you need lots of this to happen. Right, you need lots of beta-galactosidase permease turning lactose into glucose. Right, so you want really efficient expression of the LAC-operon. If you don't have much glucose and you have lactose present. Right, so this kind of gets back to the extreme that I posted up here when glucose is absent and dang it, I can't find it. You can't see the chat box while I can see it, unfortunately. Yeah, when you have glucose absent and lactose present, you have massive amounts of LAC-operon expression. Right, that's the one extreme. So this is just the thing that's being detected. to be able to infer how much glucose is present. The detection part is this process here, the catabolite activated protein, and how well those two together allow RNA pol to transcribe the lac operon. That help?
Yeah.
Okay. Again, so what might help actually is showing you the opposite, right? To kind of get the whole picture. And so essentially, when glucose is high, right? So now we've got glucose being produced, right? I actually can't remember what these little arrows are here for. No. When we've got lots of glucose now because the lac operon has been expressed, now glucose goes up and cyclic AMP goes down. And that glucose levels could be because the lac operon's doing its job, or the product of turning lactose into glucose. Could be that the bacteria is not using as much glucose. Could be that there's now actually some glucose present in the environment. All of those, it doesn't matter. It's all about glucose levels. Right, and so when there's less cyclic AMP, there's not very much bound to the cap protein. So low cyclic AMP levels equals little cap equals not very, that's kind of clunky way, efficient binding of RNA pol to LAC-P equals very little expression of the lac operon. Not zero, but very low. The way you get zero is if LAC-I was bound to the operator. So lactose is still present. You still want to be able to transcribe the lac operon. You just don't need to do very much of it. Right? Because it's done such a good job, you actually have the product that you want, that it produces. Now I'm going to get rid of these. I'm actually just going to go steal my wife's coffee. I'll make her another one when I finish class and give you a little bit of time to kind of cogitate over this and then see if you have any questions when I go back. It only take a couple of minutes. And then if we have time, we'll move on to the trip operon. But I really want you to get this as best as you can before we move on to the trip operon, because the trip operon, yeah, it's a lot more. Okay, so back in a second. No, I still can't believe I went through a big chunk of my life without drinking coffee. Okay, anybody got questions before we move on? This will take quite a bit of work to get straight for some of you. Some of you, it's going to make a lot of sense. Some of you, it's going to make a lot less. So work through this, get people to quiz you on the different like scenarios. What if we have this and this? What do we get, right? How does this equal this, right? Because these two mechanisms, or two operons, are going to together constitute, I'd probably say, about 2/5 of your exam, your last exam, right? So there's going to be a lot of questions on how these things work. Some just detail stuff. What does beta galactosidase do? Turns lactose into galactose and glucose. Easy peasy, right? There'll be other ones which like, I've got this and this. What do I get in terms of lac operon expression, right? So you need to understand this to be able to answer those harder questions, right? You're not necessarily going to be able to get it easily from just looking at the PowerPoint. Oh, dang, that's hot. Maybe that was a little over optimistic, snagging that.
Dr.
Okay.
Cook?
Yeah.
I have a question. Okay, so looking at this graph, when you talk about high levels of glucose, that's after the first process has happened, or this is showing basically two different processes based on if you're going to have high glucose or a low glucose?
So the
Or-
first process, are you referring to this?
No,
Okay.
it's the slide that we were on.
This one.
That. Mm-hmm.
Okay. So when you talk about the first process, are you talking about cyclic AMP? What bit are you
Okay,
referring to?
Yes, that. So, because when you said it talks about having glucose low, so it takes the CAMP plus the RNA polymerase to make more of that, or like I think I'm confused between.
So, I wish I had like a little glucose thing. Essentially, how can I? All of this part here. Let's see if I can. And badgers, get out of it. Here we go. All of this part is the mechanism by which glucose is detected. Right, so essentially it works as the opposite. When you have little glucose, this works really well. You've got lots of cyclic AMP, you've got lots of the catabolite activator protein, so you get lots of transcription of the lac operon. And so if you think about it in terms of the context, right? Let's see if I can draw another shape. If you don't have that, right? You want loads of this produced, right?
Mm-hmm.
You know, you want lots of beta galactosidase and the permease and the transacetylase because you need to turn lactose into glucose. Right. So you have to be able to detect the fact that you don't have glucose, right? Because otherwise you're just guessing, essentially.
And that
so
would be in the
go on.
circle, and that would be in like the area you have circled, right?
Exactly.
Okay.
Yeah.
So that those components is what is telling those receptors, hey, we need to produce more of this to produce more glucose.
Correct.
Because of, okay, got it. Okay.
So this is the feedback mechanism, essentially. So if we're making this stuff and we still don't have much glucose, then we need to keep making more proteins to turn lactose into glucose. That's part of the, you know, crank it out kind of as quick as you can deal. So then when that starts working, right? Because eventually, you know, if you produce enough proteins that can turn lactose into glucose, you'll end up having lots of glucose, right? So now, let's see if I can. Let's get rid of that weird arrow. I'm not really sure what that's there for. Now, when glucose levels increase, cyclic AMP levels decrease. So again, we still have the same mechanism, but now it's just not going to work very well because there's not much cyclic AMP. And if this doesn't work very well, then the RNA polymerase isn't going to work very well. So we're going to get much less transcription.
Makes more sense now. I
Yep.
thought these were happening not in conjunction, like one would happen. Do you know what I mean? Like it wasn't.
Yeah.
it-
Yeah, they're
okay,
totally
so-
linked together. It's very much a feedback loop, right? It's kind of
Okay.
going, you know, have I done enough? Have I done enough? No,
Mhm.
no. Oh, I have. Oh, no, I haven't. You know, it's kind of a constant monitoring essentially. So for example, you know, when we have very little transcription, there's very little lac operon expression, and you know, these proteins might get like worn out or whatever, then essentially we're going to get back to the same deal where, you know, we don't have enough glucose. And now we get back to needing more lac operon expression, right? So we have little glucose because maybe it's all been used up and we're not making any more, right? So cyclic AMP levels go up. Still have lactose present, remember. If we don't have lactose present, none of this matters, right? Because if lactose is absent, LAC-O is going to be occupied by the repressor, LAC-I. And so all of this is shut off. Doesn't matter how much glucose is present or not, it's irrelevant. There's no lactose present. This whole slide, this whole process isn't going to happen, right? So you got to keep that in mind too. Right, that's the big, you know, switch on off or door open shut, whatever you want to use. But if lactose is present, then we need to figure out how much glucose is present to determine how much lac operon expression we need. Does that help?
Okay, just to make sure.
Mhm.
This whole process, you're going to need your lac operon.
Mm-hmm.
Then you're going to need your C-AMP. You're going to need that to bind with RNA polymerase.
Well,
In order
bind to the catabolite activator protein first. Right. So
Okay,
these,
yes, the complex. Okay.
Mhm.
And then it's going to trickle down and it's going to use the enzymes to produce lactose, resulting
Whoa.
into
to turn lactose into,
glucose.
correct.
Okay, and then once you get, or it feels that it gets enough, that it's plump and happy,
Uh-huh.
then it stops there.
It turned the dial down, right? It's like, you know,
Okay.
if you're boiling a pot of water,
Got it.
right? Once it starts boiling, you just turn it down to maintain a simmer.
Okay, good Lord. And then if it feels that it's not happy and plump, then, or if it does, then it goes, it lowers it and it's basically, it has little transcription.
Right. If it has enough glucose and it doesn't
Mhm.
need a lot of lac operon expression, then there'll be little cyclic AMP and you'll get very little transcription.
Okay, understand.
Yeah.
Thank you.
No worries. Absolutely. Yeah, so in a way, it's very elegant, right? Because it can modulate how much of this it does based on what it produces. Right? It's a very cool way of doing exactly as much as you need to, but not any more than that. Now the trip operon. Ooh, the trip operon is a doozy. If any of you know what that means. Actually, it's funny because this must be like an East Coast word. Because a lot of people in Texas don't know what a doozy is. It's like a, what would be a doozy? Tricky thing, I guess. Something difficult. It's a doozy. Now, we've got the opposite type of deal here, right? Remember the context. In the lac operon, we don't need the lac operon to be expressed if there's no lactose present, right? Completely pointless transcribing it if there isn't any lactose around to metabolize. The trip operon is the opposite. And trip that we're referring to is all about synthesis of tryptophan. And that's required for translation, protein production. And so there's, again, two things going on here. One is if you're making proteins, you need tryptophan, right? So tryptophan is something that you always need because it's an amino acid, right? You can't make most proteins without it. So now we have a complete opposite context to the lac operon. This is something that we need on all the time, but we need to be able to turn it off when we don't need it. And so this is not an inducible system. This is a repressible system. And I'll type this down because this is important to get to. I mean, it's here in the PowerPoint, but it doesn't hurt to type it out too. And it's on unless it's told or unless it's turned off. And for the lac operon, the thing that turns it on is the substrate. The thing that turns it off is the product. In the trip operon, the thing that turns it off is also the product. And that's tryptophan. Right, so there's the same deal again here. There's a feedback loop. How it works is a little bit different, right? There's actually a couple of wrinkles here, which takes some kind of getting used to, believe me. And in fact, I still have really bad memories of the first time I ever taught genetics. I'll tell you, this was back in 2014. Yeah, fall of 2014. It was such a bad experience, I can recall it like it was yesterday. And talking about the trip operon was one of those. So it's not trivial to get. And there's a few different ways of talking about it. We'll probably cover most of them because it takes some time to go through. Right. Okay, hopefully I'm not scaring you too much, but oh boy. That is also, funny enough, that was when I learned not to read my student feedback forms before Christmas. It's better to leave them till after Christmas, because then if they suck, you're not going to have a terrible Christmas. That was really not very good. But anyway, formative experience, right? I'm where I am now because of the experiences I have had. Anyway, so trip operon also consists of a bunch of structural genes, right? There's 5 of them. We do. Right, so done in reverse order for some reason. A, B, C, D, and E. Right, these are all involved in the different steps of turning, I think it's charismate, whatever that is, into tryptophan. Right, so these are all enzymes that are involved in each separate step along the path to making tryptophan. Now we have a promoter, TRPP, and we have an operator called TRPO, right? And so far, so similar, right? We also have a repressor, in this case, it's called TRPR. Yeah, there we go. Just making sure. And that's encoded by a gene somewhere else. So in this case, LAC-I was the repressor. Now TRPR is the APO repressor of the trip operon. Okay, so far so good. Now we also have, and this is where things get crazy, we have something called the leader region. We'll get to that next class. We're not going to start it today because that's tricky. And that's involved in what's called attenuation or cutting short, I think is the best description of that word. So again, as with the LAC operon, we've got a big on-off switch, like so, and we've got a fine tuning. Right? TRPR is involved in is the on-off switch. Of the trip operon. TRPL equals the fine tuning. And that, now remember, I was back in the day, it seemed like years ago now, when we were talking about transcription and translation. One of the key differences with prokaryotes is that transcription and translation are coupled. This is the principle that is used in attenuation. And if it's all about translation, what is tryptophan used in? Translation. Right? So we're actually going to use translation as a means of controlling trip operon expression or transcription. Yeah, I'll leave that for now. I kind of put a worm in your ear. We'll come back to that next class. And so the simple part is TRPR, right? The on-off switch. So TRPR and no tryptophan, tryptophan equals no trip R binding. And remember, again, if nothing is bound to a prokaryotic operator, you get transcription. You know, in the case of lac operon, it might not be very good transcription in the absence of CAP-CAMP, but you still get transcription, right? There's nothing physically blocking RNA polymerase from transcribing the operon. Right? Now, if you don't have tryptophan, which is not good, right? Because that means you can't translate proteins. You want a whole load of trip-operon expression, right? But now, when you get a lot of that, you're going to make tryptophan. And so tryptophan acts as the co-repressor for TRPR. So lots of, trip equals TRPR trip, which is now a repressor. And that binds to the TRPO operator and shuts down transcription. So that's the negative feedback loop. And that's where we'll leave it, because there's no way I'm going to start talking about attenuation with a minute left. That's just not going to happen. Okay, so we'll go back over this. We'll have time next class to go over the lac operon. Make sure that all makes sense. I think we did a pretty good job of it today, but there may be, extra polish required. If you need any clarification, bring those questions to beginner class Tuesday. And we'll also quickly go back over this before we get to attenuation. Because attenuation is really cool, because it ties together a bunch of stuff that we've been learning about. Transcription and translation being coupled in bacteria. This is a fabulous example of that. And also how RNA can form multiple secondary structures by binding to itself, which we've also talked about a bunch in terms of, just RNA in general, but also those termination loops that we talked about in intrinsic termination and the like. It's kind of neat. Okay, that's enough from me. Enjoy the rest of your day. Hope you have a lovely weekend. And I'm going to finish drinking my wife's coffee. All right, take care, everybody.